Welcome to Learn While Sleeping. This is the podcast where you can drift into rest while your mind explores the quiet wonders of science. Each episode is a calm lesson, slow and steady, meant to teach and soothe at the same time. You don't need to stay awake to follow along. Your body can rest. Your thoughts can soften, and the science will simply flow around you. If this is your first time listening, you are invited to let go of the day. Find a comfortable position. Let your shoulders settle. Take a slow, deep breath in, and gently release it. Here, learning is not stressful. It is not rushed. It is gentle, like reading by the candlelight, or listening to a quiet story by the fire. Tonight, we are returning to the world within, the world of molecular biology. In our first episode, we took an overview of DNA, RNA, proteins, and energy. In this episode, we will go deeper. We'll explore how DNA is packaged, how it is copied and repaired, how RNA is processed, how proteins are made and regulated, and how these processes connect medicine, biotechnology, and even everyday life. As you listen, you may drift in and out. That's fine. There's no quiz, no test, just a calm journey inward, into the molecules that make life possible. So, settle in, breathe deeply, and let's begin. Tonight, we are going to go deeper. In our last journey, we met DNA, the library of life. But tonight, we look more closely at how that library is organized, and how its shelves are opened and closed. We'll explore how DNA is packaged into chromatin, how it folds into higher structures, and how small chemical marks, called epigenetic modifications, help decide which genes are silent and which ones sing. DNA is long, very long. If stretched out, the DNA from just one human cell would reach about 2 meters. Yet it must fit inside a nucleus less than 1/100th of a millimeter wide. How does the cell solve this? Through careful packaging. The first level of packaging is the nucleosome. A nucleosome is a segment of DNA wrapped around a core of proteins called histones. Imagine winding through that thread around a spool. That is the DNA wrapping around histones. Each nucleosome holds about 147 base pairs of DNA, wrapped around almost twice around a histone octamer. Between nucleosomes, short stretches of DNA connects them, like beads on a string. This string of beads is the foundation of chromatin. But even beads on a string are still too long, so chromatin folds further. Nucleosomes pack together, forming thick fibers. These fibers loop and coil, anchored by scaffold proteins, until they form entire chromosomes. When the cell is not dividing, chromatin exists in 2 main states: euchromatin, loosely packed, accessible, active, and heterochromatin, tightly packed, inaccessible, silent. The balance between these 2 states helps determine which genes can be used at a given time. Histones are more than spools. They are the gatekeepers. They can be chemically modified to influence whether DNA around them is open or closed. For example, acetylation of histone tails loosens the grip on DNA, making the region more open, and often leading to gene activation. Another thing called methylation can either activate or silence genes, depending on the context. Phosphorylation and other marks add more layers of control. So remember, acetylation loosens the grip on DNA. Methylation activates or silences genes. And phosphorylation adds more layers of control. These modifications are reversible. Enzymes add and remove them like editors marking the margin of a book. Together, they form a histone code, a layer of regulation beyond the DNA sequence itself. This brings us to epigenetics. Epigenetics refers to heritable changes in gene expression that do not involve changes in the DNA sequence. The DNA letters A, T, C, G remain the same, but chemical tags can be added or removed, changing how genes are read. One common epigenetic mark is DNA methylation. Here, a methyl group, or carbon with 3 hydrogens attached, is attached to cytosine bases, usually at regions called CPG islands. Heavy methylation often silences genes. Epigenetic changes can persist through cell divisions. They can even be influenced by environment, nutrition, stress, and experience. This means your genes are not your only destiny. They are also shaped by how your cells are marked and how those marks are passed along. Let's think about some examples of epigenetics. One famous example is X chromosome inactivation. In female mammals, one of the 2 X chromosomes is largely silenced through epigenetic mechanisms. This balances gene expression between males and females. Another common example is cell differentiation. Every cell in your body contains the same DNA, yet a neuron looks and acts very different from a muscle cell. This difference arises from epigenetic programming. Some genes turned on, others kept silent. Epigenetics also plays a role in memory, development, aging, and disease. Beyond chemical marks, cells use chromatin remodeling complexes. These are protein machines that slide nucleosomes, remove them, or restructure them. By moving nucleosomes around, these complexes expose or hide DNA regions, allowing transcription factors to bind or keep them out. It's a dynamic system, not a static shelf of books, but a library that constantly rearranges itself, bringing certain volumes to the front and shelving others in the back. As you rest here now, your cells are not only storing DNA, they are actively shaping it, folding, modifying, tagging, remodeling, deciding what should be read and what should remain silent. This quiet editing and arrangement is the foundation of life's flexibility. Chromatin and epigenetics are the bridge between the stable code of DNA and the ever-changing needs of the cell. DNA is a stable molecule, but for life to continue, it must be copied. Every time a cell divides, its DNA must be replicated, so that each daughter cell receives a complete set of instructions. This copying must be precise. Errors threaten the integrity of the genome, and yet the process is remarkably efficient, guided by specialized enzymes and strict checkpoints. Let's walk through the DNA replication and the systems of repair that safeguard our genetic material. Let's talk about the replication fork. Replication begins at special sites on the DNA, called origins of replication. Here, proteins open the helix, creating a bubble. At each end of the bubble, a replication fork forms. Picture it as a Y-shaped structure, where the double helix is unwound and new strands are being synthesized. The enzyme helicase unwinds the DNA, breaking hydrogen bonds between bases. Single-strand binding proteins stabilize the unwound strands. Ahead of the fork, topoisomerase relieves tension, preventing the DNA from twisting too tightly. At the fork itself, a new strand begins to form. There are leading and lagging strands. DNA polymerase, the enzyme that builds new DNA, can only add nucleotides in one direction. This is from 5' to 3'. But DNA's two strands run in opposite directions. This creates a problem. On one strand, the leading strand, synthesis is continuous. Polymerase follows the fork, adding nucleotides smoothly. On the other strand, the lagging strand, synthesis must occur in fragments. Small pieces called Okazaki fragments are laid down one at a time. Later, another enzyme, DNA ligase, joins them in a continuous strand. The result, both strands are copied, but by different strategies. There are primers and polymerases. DNA polymerase cannot start from scratch. It needs a short piece of RNA as a primer. These primers are made by an enzyme called primase. Once in place, polymerase extends the strand, adding complementary nucleotides, A with T, C with G. In eukaryotes, several DNA polymerases exist, each with specialized roles. Some extend the strands, others remove primers, others proofread and repair. Together, they create a perfect copy. But even the best polymerase can make mistakes. Some polymerases have a built-in proofreading function. If the wrong base is added, the enzyme senses the mismatch, removes it, and replaces it with the correct one. This proofreading dramatically improves our accuracy. Replication errors are rare, about 1 in 10 million nucleotides, and even those errors can often be corrected later. At the ends of linear chromosomes lies a special challenge. When the lagging strand is replicated, the very end cannot be copied. This would cause chromosomes to shorten with each division. To solve this, cells use what's called a telomere, repetitive, non-coding DNA sequences that act as protective caps. Over time, telomeres do shorten. This shortening is linked to aging, but an enzyme called telomerase can extend telomeres, adding repeats to the ends. Telomerase is active in germ cells and stem cells, and often in cancer cells, helping them divide indefinitely. DNA faces constant threats: UV light, radiation, chemicals, even reactive molecules within the cell itself. Damage can take many forms: single base errors, mismatches, bulky distortions, or dangerous double-strand breaks. To protect the genome, cells use a very sophisticated multiple repair system. We have mismatch repair, which corrects errors missed during replication. There is base excision repair. It removes and replaces damaged bases. Nucleotide excision repair removes bulky lesions, like those caused by UV-induced thymine dimers. And double-strand break repair uses recombination or ligation to restore chromosome integrity. These systems act consistently, keeping the code intact. But when the repair fails, then what happens? If damage is not corrected, mutations can occur. Some mutations are harmless. Others can disrupt proteins. Still others can activate oncogenes or disable tumor suppressors, leading to cancer. Diseases like xeroderma pigmentosum arise from defects in repair pathways, making patients extremely sensitive to UV light. Repair is not something that's optional, it's essential. As you lie here now, Every cell in your body is copying DNA, proofreading it, and repairing any errors. Helicases unwind, polymerases extend, ligases seal, repair proteins patrol, correcting mistakes. And this is the quiet maintenance of your genetic library. Now we will discuss transcription and RNA processing. We've seen how DNA is stored and repaired. Now we turn to the next great process, transcription. Transcription is how the information in DNA is copied into RNA, the molecule that carries instructions outward. This is more than just copying. It is highly regulated, carefully controlled, and often fine-tuned by the processing steps before the message is ready to be used. It all starts with RNA polymerases. The central player in transcription is RNA polymerase. In bacteria, there's just one RNA polymerase, responsible for making all types of RNA. However, in eukaryotes, the most common animal cell in our bodies, RNA polymerase 1 makes ribosomal RNA, or rRNA. RNA polymerase 2 makes messenger RNA, or mRNA. And RNA polymerase 3 makes transfer RNA, or tRNA, as well as some small RNAs. So remember, polymerase 1, rRNA, polymerase 2, mRNA, polymerase 3, tRNA. I'll repeat that one more time. Polymerase 1, rRNA, polymerase 2 makes messenger RNA, and finally, polymerase 3 makes transfer RNA. Each has its own role, its own set of promoters, and its own regulation. Now, every gene has a promoter, a stretch of DNA, a sequence, you can say, that tells RNA polymerase where to begin. In bacteria, a common promoter sequence includes the 10 and 35 regions, recognized by a protein called sigma factor. However, what we are mostly concerned with is eukaryotes. In a eukaryote, one well-known promoter element is known as the Tata box, as a TATA. Often about 25 bases upstream of the start site. Promoters are like start signals, signposts that guide polymerase to the right spot. But polymerase cannot act alone. It requires what's known as a transcription factor. These are little proteins that help it bind. They unwind the DNA and start building the RNA strand. Once assembled, polymerase begins elongation, sliding along DNA, adding complementary bases. Let's talk about elongation and termination. As RNA polymerase moves forward, it unwinds a short stretch of DNA, reads one strand, and builds an RNA copy. The RNA grows base by base, A with U, adenine for uracil, and then C with G, cytosine for guanine. Behind the enzyme, the DNA helix forms. It reforms ahead of it, more is unwound. Eventually, the enzyme reaches a termination signal. In bacteria, this may be a hairpin loop in the RNA, or a protein called Rho, RHO, that stops polymerase. But in eukaryotes, termination is more complex, often involving cutting the transcript at a specific site downstream. Let's talk about RNA processing in eukaryotes. In bacteria, RNA is often ready to use immediately. But in eukaryotes, messenger RNA must undergo extensive processing before it can be translated. 3 major modifications occur. The first one is known as 5' capping. It's a special modified guanine nucleotide that's added to the beginning. The cap protects RNA from degradation and helps ribosomes recognize it. Splicing. The second modification is splicing. Genes in eukaryotes are often interrupted by non-coding sequences called introns. These introns are removed, and coding sequences called exons are joined together. The process is carried out by the spliceosome, a complex of proteins and RNAs. Sometimes, splicing can occur in different ways, often producing alternative transcripts from the same gene. And finally, the last major modification is the poly A tail. At the 3' end, a string of adenine bases is added. This poly A tail stabilizes the RNA and helps with export from the nucleus. So just to review, we have 3 major modifications before messenger RNA can be translated. 5' capping, guanine nucleotide added to the beginning. Splicing with the spliceosome, introns are removed and exons are joined together. And finally, a poly A tail. At the 3' end, a string of adenine bases is added. Now, the RNA is ready for export from the nucleus. Not all RNA becomes protein. Some RNAs play regulatory roles. MicroRNAs or miRNAs and small interfering RNAs or siRNAs can bind to messenger RNAs, guiding them to degradation or preventing their translation. This layer of control is so powerful, it allows cells to fine-tune gene expression, silencing some messages and amplifying others. There are differences between prokaryotes and eukaryotes. In bacteria, transcription and translation are coupled. Ribosomes can begin translating messenger RNA while it is still being made. In eukaryotes, transcription occurs in the nucleus. Translation occurs in the cytoplasm. The messenger RNA is exported out of the nucleus so that translation can begin there. Finally, processing steps add another layer of regulation. This separation of space and steps gives eukaryotes more complexity and flexibility. Now let's reflect on this. As you listen to this, your own cells are transcribing DNA into RNA. Imagine that. Polymerases slide along genes, building messages, capping, splicing, and tailing, preparing them for export. Other RNAs float nearby, regulating, silencing, and adjusting. Section 4. Translation and regulation. We have followed the flow of information from DNA to RNA. Now we arrive at the next step, translation. This is where the genetic code is converted into proteins, the builders and workers of life. Translation is a process of remarkable precision, carried out by some of the most intricate machines in biology, the ribosome. Let's explore how ribosomes read RNA, how amino acids are strung together, and how cells regulate this essential process. The ribosome is a molecular machine. It's enormous by molecular standards. It is made of both proteins and ribosomal RNA. In prokaryotes, ribosomes are smaller, with a 30S and a 50S subunit that come together to form a 70S ribosome. In eukaryotes, ribosomes are larger, with 40S and 60S subunits, forming an 80S ribosome. Despite the differences, the basic function is the same: to read messenger RNA and assemble amino acids into proteins. The messenger RNA carries instructions written in groups of 3 bases, called codons. Each codon specifies an amino acid. For example, AUG codes for methionine and also serves as the start codon. UUU codes for phenylalanine, and GGC codes for glycine. There are 64 codons in total. 61 specify amino acids. 3 serve as stop codons, telling the ribosome where to end. This code is nearly universal, from bacteria to humans. The same codons specify the same amino acids. There are charts that you can find on Google or anywhere else where you can figure out exactly what codon corresponds to the 3 base pairs. Let's think about transfer RNA. To match codons with amino acids, cells use transfer RNA or tRNA. Each transfer RNA has 2 ends. One end carries a specific amino acid. The other end carries an anticodon that pairs with the codon on mRNA. When the codon and the anticodon match, the ribosome links the amino acid to the growing protein chain. This process repeats codon by codon, amino acid by amino acid, until the full protein is built. There are 3 stages for translation. The first stage is initiation, and the second one is elongation, and the last one is termination. In initiation, the small ribosomal subunit binds to mRNA. The start codon, AUG, is recognized. The initiator, which is transfer RNA, carrying methionine, binds. The large subunit joins, forming a complete ribosome. In elongation, the new ribosome moves along the messenger RNA. New transfer RNAs bring amino acids, one codon at a time. Now a peptide bond is forming, linking amino acids together. This protein grows longer, folding as it emerges. Finally, in termination, a stop codon is reached. Release factors will bind, causing the ribosome to let go of the protein. The ribosome disassembles, ready to be used again. Let's review those 3 steps. Initiation, elongation, and termination. In initiation, the ribosomal subunit binds to mRNA. AUG is recognized, and it binds with the initiator, tRNA. In elongation, peptide bonds form, linking amino acids together. The protein is growing in this stage. In termination, a stop codon is reached, and the ribosome can let go of the protein. There's a codon bias and efficiency. Not all codons are used equally. This is called codon bias. Some codons for the same amino acid are used more frequently than others. This bias affects how efficiently proteins are made. Different organisms have different codon preferences. This is important in biotechnology, when expressing genes across species. How do we regulate translation? Cells carefully regulate this via initiation control, which is the decision to start translation, and it's often the key point of regulation. Another way that cells regulate translation is by RNA binding proteins. These binding proteins can block ribosome binding or stabilize the RNA for use. A third way is microRNA. These small RNAs can silence messenger RNAs, preventing translation altogether. Finally, there's global regulation. When you're really stressed, cells may reduce overall translation to conserve energy. This flexibility allows cells to respond quickly to conditions. You can see that there is a variety of mechanisms to regulate translation. A classic example that we should discuss is the lac operon. In bacteria, one of the most studied systems is the lac operon, or LAC. The lac operon contains genes for breaking down lactose, a sugar. You see, when lactose is absent, a repressor protein binds the DNA, blocking transcription and translation. When this repressor protein binds the DNA and it blocks transcription and translation, this is what happens. Lactose, whenever it's present, it inactivates the repressor. The genes are expressed, enzymes are made, and lactose can now be digested. This operon demonstrates how cells turn genes on and off based on need. It's a simple, elegant example of gene regulation. As you rest tonight, your ribosomes are at work. They read messages codon by codon. They also string amino acids together, elongating them. They build enzymes, hormones, antibodies, receptors, proteins that fold and function, sustaining your life. the translation of information into form, the translation of code into matter. Section 5. Molecular Biology Techniques. So far, we've looked inside the cell, at chromatin, replication, transcription, and translation. But how do we as scientists explore these processes? How do we see what DNA is doing, or measure what proteins are made of, or edit the code of life itself? For this, we use tools, instruments of molecular biology. How about we take a slow walk through the laboratory and imagine how these techniques open the cell's world. The first thing we're going to talk about is PCR, and it's how we copy DNA. Imagine standing at a small bench. Before you sits a slim plastic tube with a tiny droplet of liquid inside. Believe it or not, inside that droplet are fragments of DNA. If you need more, let's say millions of copies, you turn to PCR, the polymerase chain reaction. By now, polymerase should be a familiar word to you. The sample is placed into a thermal cycler. The sample then heats, denatures the DNA strands, then cools, allowing primers to bind. With this heat and denaturation of the DNA, the primers bind and a heat-stable enzyme called Taq polymerase, or TAQ, extends the primers, adding nucleotides. This cycle will repeat, heating, cooling, and extending, over and over again. Heating, cooling, and extending. Heating, cooling, and extending. Heating, cooling, extending. Each round doubles the DNA, from a single piece to billions. In just a few hours, you have enough DNA to study, to sequence, to clone. A simple reaction, but one that revolutionized biology. Let's also talk about gel electrophosphoresis. Don't be alarmed by the name. This is how we can see DNA. Imagine sliding your DNA sample into a soft gel, like a clear rubbery cushion. The gel is placed in a box of buffer, and an electric field is applied. DNA, which is negatively charged, begins to migrate towards the positive pole. It's attracted to the positive pole and it goes there slowly. Smaller fragments move faster, slipping through the gel. The larger fragments, they lag behind. Why would that be? It's because of the size of the fragments would have to do with their ability to reach the negative side faster or slow. The side would affect their speed. After a while, you shine light across the gel. Bands appear, each one a size of DNA. It's like seeing the invisible. Something that's invisible which is made visible in glowing stripes. How about reading the code? Once you have DNA, you may want to read it to know the exact order of bases, A, T, C, and G. The earliest method of this was called Sanger sequencing. It uses special nucleotides that stop DNA synthesis. By running many reactions, scientists piece together the sequence. Today, newer methods, like next-generation sequences, read millions of fragments at once. Machines, they hum quietly, analyzing signals, turning them into digital code. On a screen, the bases scroll past. A, T, G, C. The letters revealed. Another technique is cloning and recombinant DNA. Suppose you want not just to read DNA, but to move it, to insert it into another organism. This is the process of cloning and recombinant DNA technology. Imagine cutting DNA. What if we wanted to cut a piece of DNA or a sequence? Well, we use something that's called a restriction enzyme. It's like a molecular scissor that recognizes a specific sequence. You just insert the fragment into a plasmid, a circular DNA molecule. The plasmid is introduced into the bacteria, which grow and divide, making copies of the inserted gene. Soon, you have a living factory producing your new DNA, or even making the protein it encodes. This is how insulin was first produced, using recombinant technology, a life-saving medicine born from bacterial cloning. The next technique is known as a blotting technique. There are a couple that we might have to discuss. Blotting techniques are for detecting molecules. To study DNA, RNA, or proteins, we use different types of blots. A southern blot detects our DNA. A northern blot detects RNA. A western blot detects proteins. Let's repeat that again. A southern blot detects RNA. A northern blot detects RNA. A western blot detects proteins. And we'll repeat that one more time. A southern blot detects DNA. A northern blot detects RNA. And finally, a western blot detects proteins. The idea is similar between these three. Separate molecules by size, transfer them to a membrane, and probe them with labeled markers. Bands will appear showing presence and abundance. These blots are like fingerprints, revealing unique patterns that reveal what lies within a sample. What about CRISPR, or CRISPR, a method for editing DNA? This is a much more contemporary technique. Finally, we can imagine a tool that not only reads DNA, but changes it. This is CRISPR-Cas9. Borrowed from bacteria, CRISPR uses a guide RNA to find a matching DNA sequence. Cas9 a protein, and it cuts the DNA at that spot. Scientists can then add or remove sequences, editing the genome directly. The possibilities for this are vast. We can use it for studying genes, developing therapies, and even reshaping the modern biology research scope itself. Let's reflect on this. As you imagine these techniques, polymerase chain reaction or PCR, copying DNA, gels in electrophosphoresis, separating fragments, sequencers reading bases, plasmids carrying genes that you cut, blots detecting signals, and CRISPR editing the code. Remember that they are tools to see the unseen, to bring the hidden world of the cell into human hands. Machines in labs reveal the deepest secrets of life. Section 6. Molecular Biology in Medicine. Molecular biology is not just theory. As much as we talk about it, it's not only just about DNA, RNA, and proteins in the abstract. These processes play out in real life. When they work well, they sustain health. When they falter, they lead to disease. Let's take a calm, relaxing walk through the medical world of molecular biology. Imagine a cell copying its DNA. As you know, helicase unwinds, polymerase builds, and proofreaders check each base. Most of the time this system is flawless, but sometimes a mistake does slip through. This is what's known as a mutation. If that mutation affects a gene that controls growth or repair, the cell may begin dividing uncontrollably. That is the beginning of cancer. For example, a mutation in the p53 gene, a guardian of the genome, removes an important checkpoint. The cell ignores signals to stop and continues to divide. Let's repeat that again. When there's a mutation in the p53 gene, it removes an important checkpoint, and then the cell ignores signals to stop and continues division. Molecular biology has revealed that cancer, at its core, is a disease of the genome, a story of damaged instructions. However, not all mutations lead to cancer. Some cause inherited disorders. Take cystic fibrosis. It is caused by a mutation in the CFTR gene. The CFTR gene is a regular gene that occurs in everyone's body. This gene encodes a protein that transports chloride ions across cell membranes. When the protein misfolds or is absent, ions cannot move properly. Mucus becomes thick and sticky. Breathing becomes difficult. Here, one change in a single gene ripples outward to affect an entire life. Another example is sickle cell anemia. A single base substitution in the hemoglobin gene changes one amino acid. This small shift causes red blood cells to take on a crescent shape. The altered cells clog vessels, causing pain and complications. These examples remind us how precise molecular biology must be. One change in one letter can alter the entire organism. That being said, not all regulation comes from mutations. Sometimes the DNA sequence is unchanged, but the way it is marked is altered. This is the realm of epigenetics. For instance, abnormal DNA methylation can silence tumor suppression genes, contributing to cancer. But stress, diet, and environment can also influence epigenetic marks. Over time, these changes may affect health and aging. Research suggests that trauma can leave epigenetic signatures that may even be passed to future generations. In this way, the environment and experience weave themselves into the molecular code without altering the letters themselves. But what about RNA and its footprint in medicine? The central dogma taught us that DNA makes RNA and RNA makes protein. But RNA itself has now become a powerful medical tool. During the COVID-19 pandemic, scientists developed mRNA vaccines, or messenger RNA vaccines. These vaccines deliver a short piece of RNA encoding part of the viral spike protein. Cells translate it into the protein, display it, and the immune system learns to recognize it. No virus is injected, only instructions. This is molecular biology in action, the language of RNA, turned into a shield for humanity. Medicine also uses molecular biology to diagnose. PCR, the method that copies DNA, was adapted for testing infections. A swab is taken, viral RNA is extracted, converted to DNA, and then amplified. If the viral sequence is present, PCR reveals it. It's amplified to such an extent that you would be able to tell if the viral sequence was present or not. This principle helped track HIV, influenza, COVID-19, and many more. Molecular diagnostics now identify not just infections, but genetic disorders, cancers, and risk factors for disease. I don't want you to worry about the diseases or possible errors. As you listen now, your cells are quietly transcribing, translating, and repairing. Most of the time, they succeed perfectly. But when these errors do happen, we do see the results in medicine, in cancer, genetic disease, epigenetic misregulation. Yet at the same time, molecular biology and its research offers hope. With PCR sequencing and RNA-based therapies, we can now detect, understand, and even correct what once seemed untouchable. It's the same science, the same molecules, seen from a new angle. The molecular biology of health and disease. Section 7. Molecular biology in biotechnology. Let's talk about forensics. Imagine a crime scene. A single hair lies on the floor. It contains only a tiny amount of DNA, far too little to analyze directly. But with PCR, the polymerase chain reaction, scientists can amplify it. Cycle after cycle, the DNA doubles. Soon there are millions of copies, enough to examine. Short tandem repeats, unique repeating sequences in the genome. They are compared across samples. If they match, the identity of the individual is revealed. This is DNA fingerprinting, and it transformed forensic science. Before biotechnology, people with diabetes relied on insulin extracted from animals, often pigs or cows. It worked, but was limited and sometimes impure. Then came recombinant DNA technology. Scientists cut the human insulin gene and inserted it into a plasmid. The plasmid was placed into bacteria. The bacteria grew, producing insulin as they divided. For the first time, human insulin could be made in large quantities, pure and safe. This was one of the earliest triumphs of molecular biology in medicine. Let's talk about agriculture. In recent years, a new tool has emerged. As we know, and we've talked about it today, CRISPR and Cas9. It uses a guide RNA to find a precise DNA sequence, and an enzyme, Cas9, to cut the DNA at that spot. With this, scientists can add, remove, or alter genetic material. In agriculture, CRISPR is used to make crops resistant to disease or to improve yield. In medicine, it's being tested to correct mutations in human genes. For example, CRISPR therapies are being developed for sickle cell anemia or for inherited eye disorders. The ability to edit DNA directly has changed the future of biology. Not all people respond to drugs the same way, though. Some medicines are effective for one patient, but ineffective or harmful for another. By sequencing individual genomes, doctors can tailor treatments. These are personalized medicine. For example, cancer tumors can be sequenced to reveal specific mutations. Those therapies can then be chosen to target those mutations directly. What was once one treatment for all is becoming one treatment for one. And it is possible because molecular biology tools let us read and understand the genome with unprecedented precision. Section 8. Molecular biology in everyday life. Imagine a summer afternoon. You step outside, and sunlight warms your skin. But along with the warmth, ultraviolet light is striking your cells. UV radiation can cause neighboring thymines and DNA to bond together, forming thymine dimers. If not repaired, these distortions interfere with replication. Mutations accumulate. This is why prolonged sun exposure increases the risk of skin cancer, and it's why sunscreen was invented and marketed by chemists like Franz Greuter in the 1930s. It remains a crucial protection for our skin health. The story of sunburn and skin health is written at the molecular level. Now think about antibiotics, medicines that fight bacterial infections. Many of them work by targeting bacterial ribosomes. For example, streptomycin, discovered by Selman Waxman and his team in the 1940s, binds to bacterial ribosomes, causing errors in protein synthesis. Human ribosomes are slightly different, so the antibiotic can harm bacteria without damaging our own cells. This principle is why antibiotics like tetracycline, erythromycin, and gentamicin remain effective tools against infections. They exploit subtle differences in the machinery of translation between prokaryotes and eukaryotes. Consider lactose intolerance. For many adults worldwide, drinking milk can cause discomfort, bloating, cramps, and indigestion. The reason lies in the enzyme lactase, which breaks down lactose into glucose and galactose. In most mammals, the gene for lactase is turned off after weaning, but in some human populations, mutations allowed the lactase gene to remain active. This persistence of lactase, sometimes called lactase persistence, spread amongst groups who practiced dairy farming. Here, culture and molecular biology intersect. A human tradition, drinking milk, led to a genetic adaptation, which is still reflected in the population today. Now think about stress, the fast pace of modern life, work, exams, responsibilities. Chronic stress increases levels of cortisol, a hormone that travels into cells and influences transcription. Studies show that prolonged stress can leave epigenetic marks on DNA, altering how genes are expressed. For example, research at McGill University in Canada revealed that childhood stress can change methylation patterns in genes related to stress response. These marks may last for years, shaping health and behavior. Molecular biology explains how experiences, intangible events in our lives, leave physical signatures on our DNA regulation. From sunscreen in the summer to antibiotics prescribed by a doctor, to a glass of milk, to the effects of stress, molecular biology is present in all these examples, and they connect to the visible events of everyday life. Section 9. Molecular Biology in the Future. So far, we've explored the machinery of life. We've seen how it connects to medicine, biotechnology, and everyday experiences. But molecular biology is not only about the present, it's also about the future, about where these discoveries may lead us. Let's take a calm look at the horizon of molecular science. Synthetic biology asks a bold question. What if we don't just study life, but design it? Researchers are already engineering bacteria to produce fuels, plastics, and medicines. In 2010, a team led by J. Craig Venter created the first synthetic bacterial genome. They assembled DNA in the lab, inserted it into a cell, and the cell came alive, guided by man-made instructions. The idea of building new forms of life from the ground up is now something that is now considerably attainable. For decades, medicine treated symptoms. If a gene was faulty, we could only address the results. Now, with gene therapy, we aim to fix the cause. By delivering healthy copies of genes or editing mutations directly, scientists are beginning to treat diseases at their roots. In 2017, the FDA approved Luxturna, a gene therapy for an inherited form of blindness. It delivers a working gene into the retina, restoring vision to patients who once lived in darkness. We've already seen how CRISPR can edit DNA with precision. Today, researchers are pushing further, developing variations, like base editors, which change single letters of DNA, and prime editors, which can rewrite sequences with even more accuracy. These tools could one day correct thousands of genetic diseases. At the same time, they do raise questions. How far should we go? Should we edit embryos? Should we change traits? Not just cure disease? That is an ethical concern. What about personalized genomics? Sequencing entire genomes is becoming faster and cheaper. What once cost billions of dollars can now be done for a few hundred. Companies like Illumina and consumer platforms like 23andMe have made genomics accessible. The future of medicine may involve doctors tailoring treatments not to the average patient, but to your exact genetic profile. Drugs, diets, therapies, all chosen for your unique biology. There's a horizon ahead. Molecular biology is moving rapidly, from synthetic organisms to gene therapies, to precision editing, to personalized care. It's a field where imagination and reality are beginning to meet, and as the tools grow sharper, the responsibility does grow heavier. What we do with this knowledge will shape the future of medicine, society, and even humanity itself. Let's do a gentle recap. We began tonight by stepping deeper into the cell. We just saw how DNA, our library of life, is not just stored but packaged into chromatin, opened and closed like shelves in a library, marked by the quiet notes of epigenetics. We then followed DNA as it was copied. We stood at the replication fork, watching helicases unwind strands, polymerases extend new ones, and repair systems patrol for errors. Next, we watched transcription, polymerases sliding along genes, transcription factors guiding them, and spliceosomes shaping the final message. From there, we walked to the ribosome, where codons were read, and tRNAs delivered amino acids one by one, until proteins emerged and folded into form. We then paused in the laboratory to see the tools of molecular biology: PCR amplifying fragments, gels separating DNA, sequencers reading bases, plasmids cloning genes, and CRISPR rewriting the code. Then we turned outward, into medicine. We saw how mutations lead to cancer, how misfolded proteins cause disease, how messenger RNA vaccines protect us. We moved into biotechnology, where PCR aids forensics. Insulin is made into bacteria, and CRISPR is shaping agriculture and medicine. And finally, we connected these ideas to daily life. Sunlight causing thymine dimers, antibiotics exploiting ribosomes, lactose intolerance rooted in genetics, stress leaving epigenetic marks. At the horizon, we glimpsed into the future. Synthetic genomes, gene therapy restoring sight, CRISPR evolving into even finer tools, and personalized medicine becoming a reality. All of these threads are part of the same tapestry, molecular biology. The science of life's smallest parts. It is precise, it is dynamic, and it is what makes us human. Because the same molecules that quietly hum inside your cells tonight are also the keys to healing, to discovery, and to the future of medicine and technology. So now, as your body rests, remember that your cells do not. DNA is guarded, RNA is written, proteins are built, energy flows, signals pass, life renews. It's all happening now. Let these quiet processes be your comfort. The orchestra of molecules plays on as you drift into sleep. Good night.